Hello, everyone. Welcome to my video tutorial for spatial dataset analysis. Today, I'm going to show you how to analyze the MERFISH mouse liver dataset using the package Squidpy. You can download the data for practice from the Vizgen website. You need to register your information in order to download this dataset. So I downloaded the data already. For today's analysis, we just need the count files, liver slice cell by gene, that's the expression CSV data file, and also the mat file. In the mat file, we have the spatial coordinators information for this dataset. So I saved both CSV files in my folder named as liver1/1. So first we need to load all the packages for today's analysis. So now we can use the read Vizgen function from the Squidpy package. We specified the file path, and we need the expression matrix data files as the count file. Also, we need the metadata file. So now we can load the data and create a data object. Okay, you can see we created a data object. In this dataset, we have nearly 400,000 cells and 347 genes. So next, we can process the data. First, we make the variable names. They are the gene names unique. After that, we can get the mitochondria gene names. Then we can calculate the quality control matrix, including the mitochondria DNA percentage. Let's calculate the quality control matrix in this step. If you compare the A data object, you can see we add more information into the A data observations and variables. So all the information were obtained by the quality control matrix calculation. So after that, we can filter the data. We are going to follow the online tutorial to keep the cells that have at least 50 count in total, and also keep the genes that at least expressed in 10 cells. So now we can filter the data. If you have looked at the observations, now we only have 360,235 cells, but we still have 347 genes. So after the data filtration, we can run the standard Scanpy workflow to analyze the data. First, we normalize the data. After that, we run log transformation. Then we scale the data. Because we only have 347 genes, so we don't need to identify the highly variable genes. So we can use all the genes to perform PCA analysis. After that, we find the neighbors, run UMAP, and we use the latent method to identify the cell clusters. Here we use resolution as 1.5. Let's run. Because we run all the steps for the Scanpy workflow, it will take some time to finish in this step. I will cut off the running time from the video tutorials. Okay, we finished the Scanpy workflow to identify the cell clusters. Now we can use UMAP plot to see the cell clusters. Because we use the latent method, we can count the cells by the cluster number in the latent column of the metadata. Let's plot the cells. You can see in total we got 25 cell canisters. Because we have the cell canisters here now, we can visualize the cell canisters on the spatial coordinators. So we can use the spatial plot function to visualize the latent cell canisters on the spatial coordinators. Let's see the canister distribution on spatial coordinators. You can see we generated the figure for spatial coordinators. You can see here we have 25 cell canisters in total. You can see distinct the spatial localization for different cell canisters. So now we have analyzed a data object. You can visualize the cell canisters, and also you can see the cell canisters on spatial coordinators. Next, we can use some mark genes to show their expression on UMAP canisters and also on spatial coordinators. For example, we can use HC and AXIN2 as haptocyte markers to visualize the haptocyte. First, on the UMAP cell canisters, we can run. You can see HC labels the haptor cells in this area. Because the AXIN2 labels haptocyte next to the pericentral blood vessels, you can see the pericentral haptocytes population is largely in this area. So we can see HC and AXIN2 expression on the cell canisters. Now we can see their expression on the spatial coordinators. Let's run. Okay, now you can see the expression of HC and AXIN2 on the spatial coordinators. Because the HC enables all the haptocytes, then you can see all the haptocytes in the liver tissue. Compared to the HC expression, AXIN2 only labels the haptocytes next to the pericentral blood vessels. Then you can see on the spatial coordinators, AXIN2 label the cell population next to the blood vessels. In next demonstration, we are going to use the blood vessels marker gene VWF to show the blood vessels. And also in this demonstration, we use the MRC1 gene to label macrophages. First, we are going to show their expression on the UMAP cell canisters again. So you can see VWF label endothelial cells in this area, then MRC1 label macrophages in this cell canister and this cell canister. So now we can realize VWF and MRC expression on the spatial coordinators. Then we can identify the blood vessels and also macrophages. Okay, you can see VWF label the blood vessels. You can clearly see the large blood vessels morphology here. Then MRC labels macrophages on the liver tissue. You can see the even distribution of macrophages in the liver tissue. So I showed you how to use the marker genes and identify different cell populations. I'm not going to demonstrate the marker gene identification and the cell type annotation in this video. If you want to identify the marker genes for your dataset and also annotate your cell types, you can watch my Scanpy video tutorial. So once we have analyzed the cell types, and also we can visualize the cell types on spatial coordinators, we can do more analysis. For example, we can perform the neighborhood enrichment test. So this test could help us to identify cells belong to different cell clusters on the UMAP cluster, but they could show neighborhood localization on the tissue. So to perform the neighborhood enrichment test, first we calculate a connectivity graph. This graph shows the cells and also cells interaction. Let's calculate the connectivity graph first. Okay, you can see the progress for the calculation. So you can see it reaches 100%. Now we can plot a heat map to show the clusters of enriched neighborhood. Let's plot the heat map. We can scroll down to see the heat map. You can see from the corner, the yellow color shows the possibility that a cluster could localize next to each other in the liver tissue. For example, you can see here class 14, 20, and 19. They could be localized next to each other on the liver tissue. So we can subset the cluster 14, 19, and 20 and create a new data. Let's subset these three cell clusters and create a new data as B data. Then first we can plot the UMAP cell clusters to see if indeed we subsetted 14, 19, and 20 cell clusters. Let's run. You can see on the UMAP plot, we only have cluster 14, 20, and 19. If we run the spatial plot now for cluster 14, 19, and 20, let's run. You can see on the spatial coordinators, class 14, 19, and 20, they show very close localization, and all the cells are around the blood vessels. So from the spatial coordinators plot, you can see we can use the neighborhood enrichment test to identify the cell clusters that show enriched neighborhood in the tissue. So this is the neighborhood enrichment test. Next, we can perform one more demonstration. We are going to perform the Moran's I-score analysis. This analysis can investigate the spatial distribution of gene expression in the tissue. So first we can use this code to perform the calculation for Moran's I-score. Then we are going to identify 12 genes for the top autocorrelation and the bottom autocorrelation. Let's run the Moran's I-score analysis. So after the analysis, we can use the spatial plot to see the gene expression on the spatial coordinators. We are going to plot genes for top autocorrelation. You can also change the color here as bottom autocorrelation to plot the bottom 12 genes. Okay, you can see we generated a spatial plot to show the top 12 genes that have high spatial autocorrelations. You can see for the top 12 genes, they enable hepatocytes in the liver and also cells associated with blood vessels. So the Moran I-score analysis indicate the primary gene expression autocorrelation is largely driven by the cells in the blood vessels and hepatocytes in the liver tissue. So this is the last demonstration. You can use the analyzed data to perform more analysis. I'm going to stop here for today's demonstration. If my video tutorial could help your data analysis, please subscribe my channel if you haven't to do so, and also share my videos with your friends. Thank you.